Hi everyone, so I am Capo, a postdoc researcher at the Institute of Marine Science at Barcelona in Spain, and I'm also a guest researcher at SLU at Uppsala in Sweden. Today I will talk about applying molecular tools to study microbial transformation of mercury in ecosystem and both in aquatic and terrestrial ecosystem. First, what is mercury transformation in the environment and what drives that? Part of the transformation of mercury is mediated by microbes that are able to transform either divalent mercury into methyl mercury or divalent mercury into volatile mercury. And those transformations are done because those microbes have in their genomes genes involved in those metabolism. For instance, they have the hgcAB genes pair that allows them to do mercury methylation. Other microbes are able to do demethylation of methyl mercury using merB gene, while sometimes the same microbes or other possess the merA gene able to do mercury reduction. These processes of mercury transformation are not only mediated by microbes. They can also be due to abiotic transformation like photodegradation in specific conditions. And this is for that you can find abiotic methyl mercury demethylation, abiotic mercury reduction. But the transformation of divalent mercury into methyl mercury is mediated by microbes, as we understand it today. When methyl mercury is present in an ecosystem, it will be bioaccumulate into food web. Here I show an example of aquatic food web, where methyl mercury is actually captured by phytoplankton species that are grazed in turn by zooplankton, etc., until the final consumer that can be sometimes human. And then when you eat a lot of fish or seafood that is contaminated with methyl mercury, you can be very sick because methyl mercury is a neurotoxin. The key question is, why are we looking for genes involved in mercury cycling? Why this is interesting? The reason why we are still looking at genes is because we don't fully understand where methyl mercury formation is happening in the ecosystem, but also the other transformation of mercury, which condition drives this process, and what is the exact role of microbes in this process. And genes are very interesting because they are the blueprint of life, and genes allow to produce proteins. So I put here the classical view of how proteins are produced by microbes. So in their genomes, genes are present and they are transformed into RNA sequence, transcript, and those RNA molecules that can actually be translated into polypeptides that are the basis of proteins. So from DNA, you will get RNA, and then you will get amino acid sequence forming the proteins. And targeting genes, we can actually know if genes present in the genome of microbes are able to be involved in a specific transformation of mercury or not, because scientists discover which genes were involved in this process. For example, for mercury methylation in 2013, Jerry Parks and colleagues from Oak Ridge National Lab discovered the gene pair named hgcAB. This is very important because before that we knew that mercury methylation was mediated by microbes, but we didn't know exactly which microbes, and we were doing inference based on the microbial diversity present in a specific ecosystem, or because of a lot of culture in the lab, some people were able to actually isolate strain able to do mercury methylation. They were mostly in three taxa that are delta proteobacteria, firmicutes, and methanogenes from archaea. And the discovery of these genes allow actually to expand the non-diversity of mercury methylator in the ecosystem. The gene hgcA and hgcAB encode for chorinoid proteins and the ferrodoxin, respectively. This discovered was done in 2013, as I said, and in this specific paper, the scientists, they actually mutate those genes to see if the strain having the genes mutated were able or not to do micromethylation. Actually, it was not the case after mutation, so it proves the role of those genes in micromethylation. Usually, in the strain isolated, hgcA and hgcB genes are found collocated in the genomes, but sometimes you can find a gene between them. So it allows a very good detection of them in environmental genomic data, because once you detect hgc genes, you can actually detect hgcB genes aside it. It seems to not be the case for all genomes. In some case, hgcB can be found in a very different part of the genomes. There is a high number of papers using this information about the genes to detect them in environmental genomic data, as I just put on the right part of this slide. What is very interesting about this gene, but also very useful for us doing bioinformatics, is the fact that some amino acids are essential for the function to happen. If you just delete them or substitute them, you will actually not have micromethylation happening anymore. And then you can detect those amino acids in the genes you find in the environment. For instance, in hgcA gene, you can find that most strains able to find micromethylation have this specific motif that is NVWCAAGK with variation of some amino acid sometimes. So using this information, and the same with hgcB that have a specific conserved motif, you can actually say, okay, this may be a hgcA gene, or this may be a hgcB gene. Because when you find those genes in water sample or sediment sample, you don't really test, you don't isolate a strain with a specific test for micromethylation. You can't say for sure this is a real gene involving micromethylation. So usually we call them hgc-like genes when they come from the environment. Over the last years, we have been working on designing a tree of life for micromethylator, let's say, using the information in hgcA and hgcB sequence. And it was started by people from Oak Ridge National Lab, Kathleen Gianfrido, and also people from Madison University in the group of Trina McMahon. We compiled all this analysis they did into a common database, a gene catalog that I will describe at the end of this talk, and we came to this specific tree, phylogenetic tree of the diversity of microorganisms carrying hgc genes. And you can see that most of the genes are actually from Desulfobacterota. It was a group named Delta Proteobacteria a few years ago. And we also identify a lot of hgc genes in Firmicutes, Nistrospirota, the PVC superphylum that is from Tomicete, Chlamydia, and Microbiota, but also quite close to Bacteriodota. Also a group of Euryarchaeota, close to Chloroflexota. Those two groups, one is Archaea and the other is Bacteria. They share similarity in the hgc genes they found. So Elizabeth McDaniels and colleagues, they suggested horizontal gene transfer between those two groups. So we have this diversity of microbes. Now I will talk about the MER operand. So it's an operand of genes that allow to do mainly two processes that are methylmercury demethylation and mercury reduction. As I said before, some microbes possess both genes, and some they just have one of them. Those genes were discovered long before hgcB genes, and quite a lot of work has been done by Tamar Barkey and colleagues on this specific microbes able to do this process, it's much more interesting because those microbes, they use this operand to detoxify their environment from mercury and from methylmercury that is quite toxic. At the opposite of micromethylation, why that is a process that we don't really understand why microbes do that actually, because it makes the product even more toxic than before. So here you can see that there is different genes involved in the regulation of this operand and also involved in transferring compound. So I will focus mostly on MER-A and MER-B genes that are the main genes involved in both processes. And using the very good dataset produced by Christos Christakis, Tamar Barkey, and Eric Boyd in 2021, we designed a phylogenetic tree, similarly to what we did with hgcAB genes, and you can see that the diversity of both microbe possessing MER-A and MER-B gene is slightly different, with quite a lot of actinobacterial genes, actually for MER-B carrying microbes. And you can see that there is a quite reasonable amount of desulfobacterota, alpha proteobacteria, and gamma proteobacteria having those genes, as well as Firmicute and Chloroflexota bacteriales. My idea for this talk was to describe the method we usually use to detect genes from environmental samples. It could be useful because this video will be online also, so even if I will go a bit quickly, you can go back to that if you want to understand something better. If I explain it good, of course. We will see about that. So basically, the molecular ecology approach is based on the fact that you go into your environment and you just collect environmental sample where you think that mercury transformation could happen, in our case. Here I illustrated by the fact that you can take water sample and sediment sample from the same environment, let's say. But you can also, of course, collect sample from soil and many other ecosystems. Extracting the DNA from specific environmental sample, you can apply to this extracted DNA two different methods to summarize. One is based on PCR amplification, and I will describe what it is, and another one is PCR-free. So this is basically sequencing all DNA fragments you can find in a specific 1 liter water sample, for example. You will apply a bioinformatics tool to actually analyze the data, the sequence data, and data analysis and data interpretation will allow you to say, okay, I found microbes carrying the genes involved in mercury transformation or not in this specific environment, and the environmental condition in which we found them. So I divide this part of the talk in two aspects, PCR-based method and PCR-free method. I will start by what is a PCR. So PCR is a polymerase chain reaction that allow actually to amplify a specific DNA fragment using primers, so molecular probes, to amplify it. The point is sometimes the DNA you will extract from a specific environmental sample is too low to be detected like that or to be sequenced, so you need to actually amplify it by PCR. And this is actually the case for AGC genes that is quite low abundant compared to other genes in microbial communities. So the principle of PCR is basically you have a double-strand DNA fragment. You will do a denaturation of this strand into two single strands using primers that are targeting specific parts of the DNA sequence. You will do hybridization, and then using a DNA polymerase, you can do the elongation of the fragment. So you will duplicate the first copy of DNA into 2 copies. This is what happened during the first cycle of the PCR, and during the second cycle, you will have a duplication of those 2 copies into 4 copies. And then doing between 25 and 30 cycles of PCR, you can actually get a lot of copies of the same genes, of the same DNA fragment, sorry. Once you get the PCR product, you can actually put that on an agar-OSIS gel and verify first if the PCR worked, and second, if the PCR works, if the fragment you amplify is at the proper size you should expect it to be. usually you will get a fragment between 100 base pair and 400 base pair when you do PCR. When I say usually, that depends a bit of your question, of course. So the take-home message about PCR, if you didn't know about it, is that PCR amplifies a specific fragment of DNA to allow a better detection of it. In some cases, you also want to sequence it, and I will explain that right later. Using a qPCR approach or ddPCR approach, you can also quantify the amount of DNA fragment you got in a given environmental sample. To do that, you will use the standard curves of DNA at different concentrations that you put in tubes in the lab that allow you to say, okay, my DNA sequence I amplify is quite low number of copy compared to the standard curves, or in the range, or a bit higher. Here I don't go in detail about qPCR, but what I think is important is Christensen, Geoff Christensen, did some paper on the fact that it may be better to use class-specific primers or hgc genes for the quantification than a universal primer. So you can read the paper I put on the right here, especially the last one from Kathleen, where she designed a bit better primer based on Geoff work. The last PCR-based method that I wanted to highlight is metabarcoding, and metabarcoding is also based on the fact that you need primer that will bind to specific region of the genome. All organisms from a same group, they have conserved region in their DNA. This is the case for bacteria, for instance. If you just look at the 16S RNA gene, some specific amino acid, some specific nucleotidic sequence are exactly the same. And between those conserved regions, you can also find variable region. And this is very interesting because if you amplify all the conserved region from different species and you actually sequence it, you can identify the different species using variable region. This is the basis of metabarcoding. So the take-home message here is conserved region in the DNA allow you to capture the DNA fragment with primers, and variable region allow you to identify different taxa. I will now go quite quickly on PCR-free method, and basically what you have to understand is you take a water sample, you extract the DNA, and you try to sequence everything from that. This is metagenomics. In your water sample, you have multiple genomes, right? Once they are sequenced, those genomes are fragmented into DNA read, so you will see the word read a lot in papers about Hg or MER genes. Reads are just short fragments of DNA. Those reads, you can assemble them into larger fragments that are named contigs, and on these contigs, you can actually predict genes using specific bioinformatic tools. And those genes, part of them, they could be the genes involved in mercury transformation. So you can count them and you can identify them by phylogeny. Once you get your genes detected with metagenomes, you can also check if you have RNA molecules or RNA fragments that map to those genes. And those RNA molecules, you will find them in the same environmental sample you get. So you extract both DNA and RNA from a water sample, you sequence both of them, and the RNA reads you get, you can actually map them to the genes that are found within metagenome. And then you can assign them, count them, and identify them. And this is very useful because you can actually maybe quantify a bit more the expression of the genes than just looking at the genes abundance. The last method that we use more and more those days is MAG reconstruction. So what is a MAG? Is a metagenome assembled genome. So basically, from the DNA reads, you assemble the read into contigs, and those contigs, they can be binned into MAGs. So you can see that you reconstruct the genome from the start with bioinformatic tools. And this is very useful because you can go a bit beyond the phylogeny of specific gene. Now you have many genes in the genome that can allow you to get a better identification of the microbes carrying the genes involved in mercury transformation. And you can also look at the other metabolic capacity. To summarize, if you use a PCR on, let's say, your AGC genes, you can be able to say if the gene is present or not. Detection then. Using qPCR, you can quantify the gene. Using metabarcoding, identify microorganisms carrying those genes. And with PCR-free method, with metagenomics, you can quantify genes and identify microorganisms carrying the genes. With metatranscriptomics, you can quantify transcripts, that is even more interesting. And finally, with microconstruction, you can better identify microbes carrying genes better than the phylogeny. And you can unravel their metabolic capacity. I just provide 2 examples of applications here. In this specific work we have been doing with Claudia, Andrea, and colleagues, we look at macrometallators or potential macrometallators in the oxic water and the anoxic sediment of Lake Geneva. And here on this correlation plot, I just put the estimate of AGC genes abundance we found with metagenomics, so MG, and qPCR. And luckily, we did that for both delta proteobacteria, so the desulfobacterota, and also Firmicutes. And when you look at MG AGCA data and qPCR AGCA data, you see a positive correlation. And same for MG AGCA Firmicute and qPCR AGCA Firmicute. Those correlations, they show that both qPCR and metagenomic estimate of AGC genes abundance are actually quite consistent between them. And this is very interesting to know that. The second example I wanted to illustrate is also a paper submitted. We also deposited on bioRxiv at the earlier this year. And here in the Baltic Sea, we got metagenomes and metatranscriptomes from 2 stations, BY32 and BY15, and at different water depths. Water depths is very interesting because we have different oxygen concentration. And what we saw here is we saw an increase of the AGC genes with decrease of oxygen, and also with increase of methylmercury concentration that I don't show here. And even more interesting, we found that there is an increase in transcript abundance in those specific water columns. This was just to illustrate 2 examples of molecular method you can use to identify microbes involved in mercury transformation in those ecosystems. Okay, I will continue and conclude this talk with building up-to-date gene catalog for AGC genes. When I started my postdoc with Eric Bjorn, my task was to actually look for macrometallators in the Baltic Sea water column, as I just showed in the previous slide. And I didn't know much about mercury and mercury genes actually. So I screened the literature, and what I realized is most of paper use slightly different protocols and slightly different database to actually detect and identify the genes. And this was an issue because it's hard to choose one of them to say, okay, these groups do a better job than these other groups. So what I did is I contacted all of them, and I said, okay, we should build a consensus database and a consensus protocol. All of them were very happy about that, so I was lucky. So I created this meta-AG working group in 2020, and we have been working since then about creating these consensus protocols. Our first outcome was the hgmate database, where we compile 2 main databases from Oak Ridge, designed by Kathleen Gianfrido, and from Madison University, designed by Elizabeth McDaniels, Ben Peterson, and colleagues. We just compiled them together and we created hgmate database this way. This database is giving a catalog of AGC genes, but also tools allowing to detect the genes and identify them, namely HMM profile for the detection and reference package for the identification, the taxonomic identification. Earlier this year, we released another preprint where we suggest a consensus protocol for the recovery of micromethylation genes from metagenomes. And this is basically a paper comparing different bioinformatics approaches in this paper we release also a pipeline named microbial that allow you from raw sequencing data to obtain an abundance of Hg, a table of Hg abundance in different environment, in different metagenomes. So we think it's very useful, and the main point of this paper is to say, okay, it's better that if you to use the same approach, all of us, it's probably not the best approach still, because it will never be the case, but at least if everyone is using the same, we can actually compare between studies, and this is the main point. Our next step is to include mere genes in the Hg-made database, and to do that we will use a very good job from Christos, Tamar and Eric, where they actually compile genomic data from online public database also together. So we want to incorporate their work into Hg-made database. I just wrote a few take-home messages. The first one is the detection of Hg cycling genes provide a meaningful information about environmental transformation of mercury in the ecosystem. This is what we show in the recent papers over the last 10 years. We need more mechanistic studies, but it will come on time. I'm sure about that. About gene quantification using either qPCR or RT-PCR, metagenomics and metatranscriptome, I think it could be useful. There is still some work also about comparison, about bias that are related to both type of method, but it may be actually useful to predict mercury transformation in the environment eventually. This is very cool that now we can actually study full genomes from microbes involved in mercury cycling. And it was the case from the paper of Daniel Jones in 2019, also Ben Peterson in 2020, and Eyulin in 2021. I think we should continue this type of work because it may allow us to really understand why mercury methylation is actually happening in the environment. And I want to conclude about the fact that collaboration is essential to actually build consensus protocols. Without that, we can't really go further about taxonomic identification of mercury methylating genes and other genes involved in mercury transformation. And if we really want to understand the role of microbes in the specific transformation, we really need to go further with building collaborative project and consensus together. I would like to thank you for listening, and thanks my three advisors, Eric, Stefan, and Andrea, and also my collaborator in the different groups I'm involved. Thank you.